So we've got about a minute to go. Does anyone have any questions before we get started about what this program is going to entail? Usually we'll be here for about 15, maybe longer, 15 minutes or longer, depending on how interested you are in what we're talking about. Do you have that amount of time? Are you?
Absolutely.
Excellent. All right. Okay. Are we ready? Are you all ready? Are you ready, Gary?
I'm ready.
Okay, so welcome officially to the California Academy of Sciences. My name is Sonia, and I'll be your presenter and interviewer today. I would like to introduce you to this chat program. This is Chat with Academy Scientists. It happens every day at 12:30, going on Thursdays, just for right now until it starts on Saturdays. And we are broadcasted to our live audience online too. So hello, live audience. Thanks for joining us. And this is Gary Williams. He is our curator of invertebrate zoology. And before we really get into that, what is an invertebrate and what is zoology? Or zoology, however you pronounce it.
Zoology is the study of animals, but there's lots of different kinds of animals, and most things that are animals don't have backbones. Only about a few percent, maybe 4 or 5% of animals have backbones like all of us. Most don't. And most of them we can't even see because they're very small, or they live in environments like the deep sea that we can't see unless we go down and investigate.
Now, I understand that you study a very, very important type of animal and somewhat well-known, the coral, a very important animal that actually creates a habitat for what they think maybe is 1 out of 4 creatures in the ocean. Why is this habitat, this coral reef, so important and so diverse?
Yeah, there's several reasons why coral reefs are so biodiverse. One is, the simple reason is because they're in the tropics, warm, sunlit waters. It's very advantageous for growth, and it's not very harsh environment. But the main reason is that they harbor algae in their tissues. The coral reef building corals have algae in their tissue. It's that algae that, through photosynthesis, gives the coral a lot of food. They can grow rapidly, and they create a lot of calcium carbonate to make these huge reefs that a lot of organisms, many organisms, can survive in.
So this is a relative, coral is a relative of things like jellies and anemones, and that algae you're talking about, it's almost as if you and I, if we had algae in our skin, we could go outside, get in the sun, and get our breakfast. So something kind of like that.
Yeah, you could drive down the street and it's lunchtime, you put your arm out the window in the sunlight, and you get plenty of food that way. It's basically the same thing.
So you get to encounter quite a few different kinds of invertebrates out there, and what is it that when you're studying them, what is it that you're trying to find out? What do you find so fascinating about them?
Well, when we go on a field trip like in the Philippines, first thing you want to do is find out what's there, what is living there, who lives there. After you make a survey, then you can ask other questions like how the biodiversity, how is it influenced in other parts of the world? How do they compare? How does one area compare to the next? Why isn't it like Antarctica or Africa or wherever else? There's biogeographic questions. How are they different evolutionary-wise? What is the evolutionary biology of different organisms? But you can't know that until you know what lives there first. So that's the basic thing we do, is go in and find out what lives there first.
Nice. And then comparing and seeing, is this different in this kind of habitat than another? Can you tell us a little bit about what we're looking at here?
Yeah, on the left is a tube anemone. These are found in shallow water in the Philippines, maybe 10 or 15 feet in depth. Most of what we do is scuba diving for shallow water, and deep water we'll get into later how we investigate that. But these are all shallow water organisms from the Philippines. That's a tube anemone. It's about 6 inches high, and it lives in a mud tube that's usually not mud colored but nicely colored, purple or red or whatever. This is a live coral with the coral polyps. These are all tentacles around the mouth of the polyp. So each polyp is a feeding individual. That's the mouth, and these are the tentacles that capture food. So these are animals that capture other animals and plants, microscopic ones, to eat.
And it's part of the color there, those algae, the little plants that live inside?
No, these are ones that do not have that.
Mm-hmm.
The color, when you see a really colorful coral, it's not from it's the coral itself. The ones that make reefs are not brightly colored, but it's the algae that forms the color.
Nice. All right. So you were talking a little bit about some of the some of the work that you've done in the Philippines. I understand you came back from an expedition last year. We went to the Philippines for how long, and why did we go to the Philippines of all places?
That was the Hearst expedition, which was a major expedition of the Academy in May and June of 2011. It's about 2 months long. And the reason why we go to the Philippines, you notice that our reef is from the Philippines, and that's based on our research locations in the Philippines. Two of us have been going there since the early 1990s. So we decided that when we built the new coral reef here at the new Academy, we were going to take what we have in our research areas in the Philippines. And the reason why we go there's two good reasons. One is it's in the middle of what we call the Coral Triangle, which is Indonesia, New Guinea, and the Philippines. And that's the most diverse area, as far as we know, in the world for marine biodiversity. So that's why we go there, because it has the highest diversity. And number two is we have a really good relationship with various agencies there for permits and for conservation agencies, education, et cetera.
So you do quite a bit of work looking for things there. When you're there, how do you conduct your research? What are you often looking for? What does the shallow water research look like?
Yeah, there's two components, the shallow water research and the deep water. The shallow water we do by scuba diving, and we go to sites that we have been told are really good at high diversity sites. And it's usually underwater photographers who have been there, or people in the Philippines from various agencies that tell us that these are really good sites, or because we've been there before, we know the good sites and the various things we're looking for. But we want a diversity of underwater habitat, and that's what on a field trip, we look for a great diversity of underwater habitat.
So when you say diversity, you mean a great number of different things. So if you're looking at numbers of different kinds of fish, numbers of different kinds of coral, that's what you mean by diversity?
And the environments are different. Like you may have a muddy environment as opposed to a really rocky nighttime environment, which is very different. So you want to see a variety of different areas when you're on a field trip.
And what are we looking at here? What are some of the creatures that you find down there?
On this last trip, we found a field or like an underwater forest of these tall, soft corals. These are all animals that look like vegetables. And there's a field at about 60 feet that just goes on as far as you can see down a slope, and they're all about 2 or 3 feet tall. So it's like an underwater forest of soft coral.
Of broccoli, perhaps, it looks like.
Broccoli, but you can notice at that depth that the colors are gone. If you bring these up to the surface, they're red and yellow and beautifully colored. But these colors are gone by the time you get down to 30 feet. Everything looks bluish.
Ooh, here's some other stuff here. This is I noticed that we were talking about those little coral algae earlier, and this is a very interesting picture here. You see a lot of color on this side, but not on the other side. What are we looking at here?
That's called coral bleaching. Everyone's heard of coral bleaching? Coral bleaching is when there's elevated sea temperatures and the algae get tired of living in that kind of environment and they leave. There's a controversy as to whether the coral expels them or they voluntarily leave. But then they go into the natural environment and they become independent bodies. They're microscopic algal cells. When they leave, what's left is just the color of the skeleton of the coral, and that's why we call it bleaching, because it's white. But that's the actual color of the coral. The brown is the color of the algae in the tissue.
And this is the hard coral you were talking about. There's some that's soft. It doesn't make that structure that's just rigid. And this is a harder coral that a lot of fish will tend to make their homes in.
The hard corals are the ones that make the reefs. The soft corals don't make the reefs. There's only 15% of all corals make coral reefs. Most corals are deeper water and off the reef, and then very deep water down to the bottom of the Mariana Trench, maybe like 20,000 feet in depth.
Now, speaking of deep water, that's a really alien habitat. That's a place that not a lot of people get to go, deep, deep down into the ocean. How do you conduct that research? What kind of tools do you use?
In the Philippines last year, we used a huge research vessel by the Philippines government. And you use deep water trawl nets that drag along the bottom, and also fish traps. So we had two ways of getting organisms. And you have a big steel cable that goes down 300 to almost 3,000 feet. And that's dragged along the bottom, this big net, and it's brought up to the surface. And then we sort through all the material on the surface in big trays. And sometimes we hit the jackpot as far as new species and all sorts of things we haven't seen before.
Yeah, and part of that Philippine expedition is going and collecting some of the specimens and bringing them back here. Now, have you ever been in a submarine? Is that what we're looking at here?
Right. This is a submarine I was in Cocos Island in Costa Rica, and this is the operator of the submarine. And we're just below the surface here. But when you get down to 200, 300 feet, it's all blue, starts getting dark, and you see some big animals. This is a big animal with a backbone. It's a ragged-toothed shark. It's about 10 feet long, right outside the submarine.
Were you a little nervous about that big shark?
As long as the submarine didn't fall apart, we were in good shape.
Now, when you were out there collecting and doing the bottom trawls, what kinds of amazing things did you find? How many specimens did you bring back?
Well, we had three components. We had a deep water, a shallow water, and a terrestrial component. So I was involved in the marine side of it. Altogether, I think we brought back thousands of specimens, and they're all here in the Academy. I personally brought back about 400 or 500 coral specimens. And when we take corals, we don't take the entire animal anymore. The days of conservation are here now, so we do like fingernail clipping, where we just take off a little piece, and the coral grows back, and we bring that back to the Academy here. There's no way you can bring back a coral this big anyway, because everything's in little tiny jars.
Wow, now I see. Is this an example of some of the methods that you use this here?
Yeah, the Filipino fisheries people were doing plankton tows to check out the surface plankton, which is not what we do. We're benthic, the bottom of the sea is what we're looking at.
Is this a good example of something benthic on the bottom?
Yeah, it's a monster from the bottom. It's a giant isopod. I think we have a specimen around here somewhere. Maybe 1.5 feet in height. It's a big isopod, and it's a voracious scavenger at the bottom, eating everything at the bottom of the sea, down to 1,000 feet or so.
Wow, very cool.
Isopod related to crabs and other crustaceans.
Now, what other cool things have you found deep, deep down in the ocean? And discovering these things, why do you think it's important? How is it important? How does it add to science?
Well, I think the reason why it's important to me is because we probably have explored the surface of Mars more than we have explored the bottom of the ocean on our own planet. So I think that we should actually learn something about here as well as anywhere else. And also the diversity. If you want to learn about overall diversity, biodiversity of the Earth, you really have to sample all the different environments. And the deep sea offers an incredible amount of environments. We don't even know about most of it. It's like scratching the surface of a iceberg.
What are we looking at here? These are some really incredible images that I've not seen too many representations of this before. What is this thing here? What is this critter?
This is only found in the deep sea, and you're not going to find it anywhere else. It's found in Antarctica, about 1,000 feet in depth. It's a 15-foot tall sea pen, a kind of coral that grows only in deep, freezing cold waters of Antarctica.
Wow, and you can see the tentacles there.
Those are the tentacles on the polyp. Those are the feeding polyps, and each one grabs food particles as they rain down from the surface. So they don't move, they're stuck in the sediment, and there's the stalk there, you can see part of it is about 15 feet long.
Wow, that's incredible. What about these things over here?
This is a deep water crab with very long legs for crawling around on a muddy bottom. And right here is a big coral. It's a Gorgonian coral. These are the kinds of corals that are not on coral reefs, so they don't have those anthellae or algae in their tissues. So they're micro predators only. They get their food by preying on other animals. They don't get it from photosynthesis.
I hear they also have some very interesting defenses, some toxins.
Some corals do. Some corals have little cells that can shoot out like a poison dart, and it grab onto little microorganisms. But most are pretty docile. They don't do a lot of damage.
Now, after going to these deep water places or finding these specimens, what kind of work do you have to do once you get back here to the Academy to help process, and what are you learning from these specimens?
Yeah, we have big laboratories downstairs for processing the animals that come in. And what we do is we curate the specimens. They come in from on the airplane. They have to be put in alcohol in jars very quickly. And then they are bottled, labeled, and put into our database. For anyone using the internet can look at our database and see what we have here. And if they want to study those animals, we can send them overseas for loans and study.
Ooh, here's some other really cool collection devices that you use on, is this a remotely operated vehicle?
That's called an ROV, which is a remotely operated vehicle. Very good. And that's in Monterey. The Monterey Bay Aquarium Research Institute has several of these. It's an underwater robot. It's tethered. It has a cable. It has fantastic cameras on it, and they're able to collect. If you can see that arm on the left, there's a hydraulic arm that can come out and actually collect. So when you're on board and you're in the control room, you can point to something and say, I want you to get a specimen of that. And it's something that's 1,000 feet or below the surface. And the operator can grab it with that arm and bring it to the surface.
Very cool. Now I understand that one thing that you did end up collecting a lot of in those deep water expeditions and those trawls happened to be plastic. Did you bring up a lot of plastic? How much did you encounter?
In the area we were in the Philippines, for some reason, I'm not sure if it's more extensive than we saw, but in that area, every trawl we had plastic and refuse, all sorts of garbage in each trawl, going down to 3,000 feet.
Wow.
And we're not sure if it's basically because of the currents in that one area or if it's everywhere. But it's a major problem worldwide. And what happens when the plastic and the garbage is thrown out to sea, it eventually sinks and goes right down to the bottom of the ocean. So we were bringing up a lot on most of the trawls.
And are there things that we as Academy scientists and also the international or local community is doing to help protect these habitats?
Right. In the Philippines, they have very good conservation organizations. And it's a critical situation because there's 104 million people in the Philippines in an area the size of Arizona. Arizona has about 6.5 million. That's 16 times the density. So everybody has to be fed and they need space to live. So it's a critical situation. That's why coral reefs are so important, because they provide food and sheltering the coastal land from storms and things like that. So they're very important for conservation. And there are good conservation organizations there, and we met with them when we were there.
And do they, what are some ways that they're helping protect these habitats?
One of the big ways right now, what we do is through science education, educating school children and the fishermen who used to use dynamite and cyanide, which are very destructive fishing techniques. That's being toned down now because of education, and they're getting the idea that they actually need to preserve their coral reef for food and shelter and that kind of thing.
Yeah, and that's part of what we do here at the Academy of Sciences. Your work has actually informed the creation of the amazing Philippine Coral Reef tank that helps educate and inspire all sorts of people from around the world. When people are looking at this amazing exhibit, what are some things that you'd like them to know about it or to notice about it?
Yeah, the interesting thing to me is that it was based on our research sites in the Philippines. And it's like 20 years of research that we came up with the idea that if we're going to make a coral reef tank, this would be a great place because we know a lot about that area. And so what you see in that tank is based on exactly what we see when we're on a field trip and diving in shallow water. The reef is only a few years old, so the corals are beginning to grow, and it will take several years for them to grow to big form that you see on the field trips that we go on. But there's a lot of different kinds of organisms, and they're trying all the time to have something else that will grow in there that they'll take and live.
Awesome. Now, anyone out there have questions for Gary? Yeah.
Yes. I know getting coral to spawn is kind of a rare event. Has it ever happened in captivity?
Oh, good question. So getting coral to spawn release its eggs and sperm is kind of tough in captivity. Have we been successful with any of that?
Yeah, we actually saw a lot of this when we were in the Philippines in nature. It happens here too. And right today there's a talk going on behind the scenes on that exact thing. The aquarium people have been doing that here. But most of what we do is we take coral samples or have them sent to us, small samples, and adhere them to a surface, like a rocky surface with epoxy glue behind the scenes here, and they grow. They'll grow into big forms. As far as spawning, we don't grow the corals from spawn here. We take samples and grow them and then put them in the tank after they've grown to a certain size.
Have we seen any of those events happen where they all spawn together?
No, in captivity, no. That's basically because of the phases of the moon. And it happened like in the Great Barrier Reef, when there's a certain moon phase, then you get everything spawning at once.
Wow.
But in here in the aquarium, it's usually foggy in San Francisco, and it's
So
a
it's a little
different
harder.
situation. Yeah.
Yeah,
You
so-
will get things that will spawn, but it's not in uniformity like you're talking about.
You never tried to recreate that event?
Yeah, we don't make
don't think we
mood
have.
lighting with the moon. We don't play certain music underwater
That's
to
a
get
good
them to
idea,
spawn.
though. I think we should
No.
try that. We need an artificial
Good
moon,
question. Very good question. Any other questions? Yeah, back here. Oh, how much diving do you do in a year?
though. Oh, that's a good question. When we go on a field trip, it's usually, well, this was 2 months, so we do 3 or 4 dives a day, which is exhausting. And so
Wow.
after 4 dives a day for 2 or 3 months, that's enough. And, but usually the field trips are less than that. They're about 2 or 3 weeks.
Do you ever go diving for fun on your own?
Not for a few years. Every time I dive, it's been for work, which
Uh-huh.
is, you can say it's basically the same thing now.
Yeah. Question over here.
Can you describe this clipping process in a little more detail? Like you say, you take little clips of the coral, and then do those become like dead specimens, or do you
So great question. So the question was, when we take clippings or specimens of coral, does that ultimately, is that going to become a sample in alcohol? Is that going to be something that can grow or it lives on?
The aquarium people will go with us on a field trip, and we're in research, which is a different division, but we work together. They come and they take live specimens back here. We don't take live specimens. We take clippings and put them in alcohol in the field, and they go in our collection. But the aquarium people will bring back live samples, put them on rocks with epoxy, and grow them behind the scenes. When they're large enough, they go into the coral reef tank. But yes, they do bring back live samples.
And here's actually a question that I often wonder about. These corals themselves, they're little animals, they grow in these colonies, but how do they grow from a small clipping to something that's much larger?
Yeah, corals are what we call clonal animals or modular animals. They're made of many parts, so you have many different feeding polyps in one individual. So it's like having 1,000 different mouths in one individual, or is it a whole colony? It depends on how you want to look at it. Is it a colony, or is it one individual with a lot of different mouths? But anyway, they all feed, and then they grow by asexual budding and just spread out over time, horizontally.
Very cool. Other questions out there? Questions in the back? Any questions from our online audience? No, maybe not today. Yes.
well, so I'm not too clear about how a coral that has photosynthesis in it is not a plant.
Ah. Okay, so how do we define coral as an animal if it has a plant living inside it that actually photosynthesizes, makes energy from the sun?
That's called symbiosis, and we are symbiotic organisms. We have bacteria in our guts, but does that make us a bacteria, or are we an animal? It's just that the bacteria is living in association with the animal, and that's what these algae do. You can take the algae out of the coral, and the coral still lives, but it won't produce coral reef. It can still survive, but it doesn't produce coral reef. But in really strong bleaching situations with the weather really bad over a long period of time, corals can die because they're not getting enough nutrients.
Yeah, so there is some food in the water that they can grab out themselves, and being an animal, eating things from outside of themselves, and that's another thing that helps us with defining them as an animal. Any other questions?
At night is when they do their feeding. The tentacles come out at night, and then the plankton is there to feed. During the day, the tentacles are withdrawn, and they photosynthesize like a plant. But they're an animal with plants living inside.
Oh, we do have a question over here from our online audience.
Have you had the opportunity to live for a prolonged time on the seafloor to do your research?
Oh, have you ever had the opportunity to live for a prolonged time on the seafloor to do your research?
No, the longest time we spent was about an hour and a half underwater in a submarine.
Okay. Well, that's a good question.
Other people have done that, though. They have lived on the bottom.
Wow. Pretty incredible. Well, what do you think? Is it pretty fun being a scientist, Gary?
Yes, it's very much fun, and you should try it one of these days, all of you.
Definitely. Do you have any words of encouragement for any up-and-coming scientists or those who are really interested in either being divers or marine biologists?
It's a matter of study, just going to school and developing an interest and being curious about the natural world. That's basically what it is.
Yeah, you can start right here at the California Academy of Sciences. Oh, one more question.
Another question, you showed the image of a lot of plastic, and this gentleman has heard about biodegradable plastics. Are they really biodegradable? Do they just break into tiny pieces? Does it make a difference?
So the question is, when talking about plastic, now we have these biodegradable plastics that are often coming up a little bit more. Are they truly safer? Do they break down in smaller pieces, or do they truly biodegrade? And
yeah, the new
make a
plastics are
difference?
supposed to do that and make a difference. What we have found are not biodegradable. These are things that from years past that have taken forever to get to the bottom of the sea, and they're not biodegradable. But the new plastics apparently are, yeah.
Do you have?
I'm not sure how they disappear or what they do. Do they break into small particles? I'm not really sure about that.
Is there a majority of a type of plastic that you've found, a type of item that you found in most of the plastic?
It seems to be plastic bags seem to be the number one.
Yeah. So it might be a very inspiring thought that pretty soon plastic bags are going to be no longer in the major stores around here. So something to look forward to out there in our city at least, and hopefully a little further around the world, because obviously it affects a lot of things all over the world. Well, can we give Gary a really big round of applause for being here and taking the time out of his research?
Thank you.
Thank you so much, Gary.
You're welcome.
Is there anything you'd like to add? Any messages for our audience here?
Maybe if you haven't seen the coral reef tank, it's just right below us here, down the stairs. It's great.
Absolutely. Thank you so much for coming here to the Academy of Sciences. Our work, like here with Gary, could not be done without any of your support. So thank you so much for being here and being curious. Continue to use your curiosity in the rest of the museum. Thanks a lot. If you'd like
Thank
to
you.
come up and ask some questions, come on over. Thanks, Gary.